I'm going to present for the lab of the course IBG213 with the title Airlift Bioreactor Operation. This experiment is to analyze the fermentation data and kinetics of the lipase production in Airlift Bioreactor. Airlift Bioreactor is the reactor that is similar to the steel-type bioreactor but without the impeller. In this experiment, we are using lurea broth as medium and the microorganism used is lipase-producing recombinant E. coli BL21 DE3. First, inoculum was prepared by taking one loop of the microorganism from agar plate and inoculated aseptically into 500 ml conical flask with 100 ml of medium and incubated at 37 degrees Celsius and 250 rpm for 24 hours. Next, lurea broth was prepared according to the formula and instruction provided. The medium was sterilized by autoclaving for 50 minutes at 15 psi and 121 degrees Celsius. The next step is the setup of the bioreactor. Setup of the Airlift bioreactor is mainly similar to the step of stir tank bioreactor. The vessel was filled with 900 ml of lurea broth and autoclaved at 121 degrees Celsius for 50 minutes at 15 psi. After autoclaving, the vessel was allowed to be cooled down. For this experiment, the temperature is set at 30 degrees Celsius and pH 7. The airflow rate was set at 100 ml per minute. After all the setting up is done, 100 ml of the culture bacteria was inoculated aseptically to the vessel through the sampling port. Media is taken as blank to measure the OD before adding the inoculum. OD of the sample is measured at 600 nanometer using spectrophotometry and recorded. Sampling is taken at different time intervals. The experiment is repeated using different airflow rates of 250, 500, and 1000 ml per minute. A standard calibration curve of dry weight against absorbance was constructed to calculate the biomass of each sample at different time intervals and airflow rate. Lipase assay was also done by mixing enzyme extract with phosphate buffer, triton X, and p-nitrophenyl laurate (PNPL) in ethanol and incubated for 37 degrees for 15 minutes. The absorbance is measured at 405 nanometer using microplate reader, and a standard calibration curve of PMP is constructed for the calculation of lipase activity in each sample at different time intervals. There are some precaution steps that need to be aware in this experiment. First, always perform regular checks of power cords for fault, fraying, or wear, and regular electrical safety checks. Besides, strictly perform all the steps aseptically, especially during media preparation, inoculation, and sampling. Another one is never fill the vessel with the media until exceeding the permitted working volume, which is normally 70% of the vessel volume. From the experiment, we can obtain the maximum enzyme activity, maximum volumetric productivity, maximum growth rate, and doubling time at different airflow rates as shown in this table. From the table, we can know that an airflow rate of 250 ml per minute, the maximum growth rate is the highest, which is 1.0165 per hour, and the doubling time is the shortest, which is 0.6819 hour. And an airflow rate of 500 ml per minute, the maximum enzyme activity and maximum volumetric productivity are the highest, which are 518.4 U per ml and 32,400 U per liter per hour respectively. Airflow rate determines the oxygenation of the fermentation process and contributes in and mixing on the fermentation broth. Airflow rate is important in an airlift bioreactor due to the absence of the impeller. It plays an important role in influencing the oxygen transfer rate, growth of cells, and productivity. From this experiment, we can observe that the maximum enzyme activity and volumetric productivity increase with the increase of airflow rate. Higher enzyme activity means that higher volumetric productivity. The enzyme activity and volumetric productivity is the lowest at 100 ml per minute and increase to the highest at 500 ml per minute. However, this started to decrease at an airflow rate of 1000 ml per minute. This may be due to the shear stress that contributed by too high of airflow rate that caused the rupture of the cells and enzyme produced facing denaturation. Cell growth has the best result at 250 ml per minute because at the airflow rate, the maximum growth rate is the highest, which is 1.0165 per hour, and the doubling time is the shortest, which is 0.6819 hour. However, higher growth rate doesn't mean that production of desired products will be higher. Thus, production of lipase is independent to the cell growth rate. In conclusion, airflow rate is an important parameter in fermentation. At high airflow rate, there is an increase in growth rate as well as in activity and productivity. However, too high airflow rate can contribute to the low enzyme activity and productivity although the growth rate is high. Hence, it is very important to choose an optimal airflow rate for the fermentation using an airlift bioreactor. In this experiment, the optimal airflow rate to obtain the maximum production of lipase is 500 ml per minute. That's all from me today. Thank you.